Hello, my name is Isabela Marques, and speaking a little about this work, mesenchymal stromal cells are multipotent, so they can differentiate into any lineage of mesoderm cells, such as adipocytes, osteocytes, etc. They have paracrine potential and are not immunogenic, making them interesting for cell therapy. And cell viability is another concept that refers to the ability of cells to remain functional in a specific environment. And for therapeutic applications such as the development of biocuratives for skin lesions, this is crucial in order to achieve right yields. However, assessing cell viability in this three-dimensional context is a challenge as the cells are incorporated into a hydrogel matrix. With this in mind, the aim was to validate the cell titer glow 3D, cell viability assay, and compare it with the Resegerin assay. Resegerin has a blue color, and after penetrating the cell in response to cell metabolism, if the cell is viable, Resegerin is reduced to resorphin, which has a pink color and a greater capacity to emit fluorescence, making it possible to monitor this reaction using fluorimetry. The fluorescence intensity is proportional to the number of viable cells. Cell titer glow 3D is an assay based on ATP, the main energy storage molecule in living beings and therefore involved in cell metabolism. Measuring the amount of intracellular ATP is an accurate way of assessing viability, as only living cells synthesize ATP. The test is based on the conversion of luciferin into oxyluciferin, catalyzed by the enzyme luciferase, together with ATP, which is a cofactor in this reaction. This ATP is released from the cell by adding a lysis buffer, and this reaction emits light of a certain wavelength. The intensity of the luminescence is measured and is proportional to the number of viable cells, since the cell is the source of ATP in this reaction. To formulate the biotides, cells derived from murine adipose tissue were combined with a solution of gelatin and alginate and bioprinted, followed by culture is in GM-EMF12, medium with and without 10% fetal bovine serum. After 48 hours, the fragments were separated using a biopsy punch for analysis with cell titer glow 3D and Resegerin, and readings were taken using the Evisible plate reader. Both assays showed that the serum-rich medium significantly increased the metabolic rate, as expected. The significance level of cell titer glow 3D was slightly greater than that of Resegerin. The results highlight the importance of considering aspects such as hydrogel composition and analytical sensitivity when selecting reagents to assess cell viability in 3D bioprinting protocols. Special thanks to CAPES, CNPq, and FAPERS.